Hi, I'm Evan Berry,
And I'm Dylan Kordic.
and we're here in Menlo Park at Pacific Biosciences to talk with Chairman and CEO Hugh Martin.
We're going to ask him all about their smart DNA sequencing technology.
Well, the way sequencing technology today in the industry works is there's some set of, or series of very complicated chemical processes, and they add one base at a time and examine one single base at a time. And the world speed champ today is a company called Illumina, and they can look at one base every 53 minutes, or in other words, one base an hour. So our idea is that instead of using a complex set of artificial chemical processes to examine the DNA, what we do is we let Mother Nature do what she does best, which, as you know, every cell eventually will divide, and somebody has to make a copy of the DNA that's in the nucleus of the cell for the new nucleus. And there's a little enzyme called a DNA polymerase. Its sole job is to replicate your DNA. So it latches onto your DNA, inspects the base that it's latched onto, and says, oh, I'm looking at a T. It says, I need the building block for a T, and it grabs that and then incorporates it into a new nascent strand that it's building. So, and then it moves on to the next base. So it's effectively sequencing. It goes base by base and looks at it. So we have a pretty simple idea. We eavesdrop on it. We watch that guy work. So we fluorescently label the 4 building blocks called nucleotides, 4 different colors, and then we get a different colored flash every time it incorporates one of those bases. And so we actually get a series of pulses, red, green, yellow, so on and so forth. And those pulses tell us, oh, that was a T, a G, a C, an A. So we go as fast as it goes and as long as it goes. So right now we're running anywhere between 1 and 3 bases per second. So that's about 20,000 times faster than Illumina. We have to build a machine. This one that we have today that I'll show you looks at 80,000 holes simultaneously, 80,000 enzymes all working in parallel, and each one of them is emitting flashes that are on the size, on the order of about 500 to 1,000 photons. If you wanted to sequence a human on any of the existing boxes today, it literally would be, if you took all of the reagents you'd have to buy in the boxes, it would fill a tractor trailer, absolutely fill a tractor trailer, to sequence a human. Whereas, you know, going back to what we do, Mother Nature sequences your entire genome with just the material in the nucleus of a cell. In other words, we use one molecule for every base we sequence. So the reason people are so excited about this technology and the cost aspect that you mentioned is the cost, the incremental cost to do a sequencing run is virtually 0. So the 3 advantages are read length, we have much longer read length, incremental cost, so it's only $100 versus $10,000, and what we call granularity, that you can run a small number of patients if you need to. You don't have to amass them all up into a huge quantity. What's unique about our company is, quite frankly, we changed everything. Every other sequencing company that exists, Illumina, Applied Biosystems, Life Technologies, 454, all of them were developed by some chemist or molecular biologist who came up with a new idea about chemistry and said, okay, great, this could work. Now, what hardware is there off the shelf, like total internal reflection microscopy or others, to see this chemical reaction? What we did from the very beginning is we said we're going to change everything. So we said we want the polymerase to free run. The minute you do that, there are a host of optical problems. It's the enzyme is only 15 nanometers on the side. So we've had to build a completely new detection system, a completely new labeling system in chemistry, and a completely new enzyme. So everything is different. So we dump all the nucleotides, all the labeled nucleotides up above and on top of this whole thing. So they're in the dark, because they're fluorescent, they only light up when the laser shines on them. They're in the dark, they're completely in the dark, because none of the light gets through the hole. Only when they get down near the enzyme do they light up. So we only see them when they're near the enzyme, because only the bottom 30 nanometers are lit up. So they're all in the dark, and then they diffuse in and out of that hole really fast, because at the nanoscale, that's what happens. And as they're diffusing in and out, they're dark, 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 and when they get down there, they're light. and they lit up, but because they're visiting really quickly, we just see a low buzz, bzz, unless the enzyme grabs one. And when it grabs it, it holds it right down there in the 30 nanometers, and it lights up, and we see this big flash.
Okay. So a much longer time.
Exactly. So in fact, the signal to noise that we get is the difference between diffusion zzz, and incorporation time.
Yeah.
Because we get a much bigger, with that incorporation pulse, we get all of the photons that are under that curve. What Dr. Jody Puglisi did over in your structural biology department, he took a ribosome, which makes proteins. He took a ribosome and put the ribosome in the bottom of a zero-mode waveguide, or a little pocket. Then he labeled 4 of the tRNAs, which are the sort of the food that the ribosome uses to make a protein. He labeled them, and we actually watched a protein being made in real time, never before seen, because you can never run a ribosome at native concentration. And so it was this paper was just published in Nature Methods, and the reviewer said this is probably one of the 10 most important papers of the year. Wow. I mean, it's a really big deal. And it's just because we can now look at a ribosome making proteins. Another thing we can do, which is a collaboration that we did with University of Texas, is we've immobilized a reverse transcriptase in the bottom of a zero-mode waveguide, and we can actually watch RNA being made in real time also. So a lot of possibilities, but Stanford, I think, is going to mainly be looking at sequencing applications.
Okay.
We're going to get to the point, probably by 2014, where you can sequence an entire human with one chip. So at that point, you can sequence a human for $100. Now, that will raise some ethical issues. Who gets sequenced, why, who gets access to that data are all going to be very interesting questions, I think, posed by the fact that you can now sequence anything. Down the road, the issues will be, there'll be ethical issues for patients, like, do you want to tell them something when there's nothing they can do about it? Then there'll be issues about what do you do if, from an insurance point of view, if you're an insurance company, what you're trying to do is maximizing your profits, and one way you could absolutely do that is if you had a completely accurate actuary table for every single customer, you could really tune your machine, which you would not want everybody to know. No one would really want an insurance company to know precisely what their genetic profile completely looked like. So all of that optics stuff you saw is sitting under here, and all that robot stuff is here. This is where you load the so these are the individual strips, and then this is where the reagents go in here, and there's the pipette tips. You load it up, and then you load it in, it takes inventory. This is just a demo, so it's not full. All of the compute is done here. So the compute resources are on here. There's like 6 quad-core fastest Intel things going. But that's it.